Okay, so hello and welcome everyone to the last discussion session of the course Fundamentals of Protein Chemistry. Today, basically, we would be discussing a few quizzes, okay, and important questions that were previously also discussed, but these questions are going to be very important for your exams and all. Okay, so before I begin, I don't see any participants at present. Anyways, we can wait for some time. But as of now, anyways, this video lecture is being recorded, so you can get to see this even later. So, so welcome everyone to the what to say the quiz session of the course Fundamentals of Protein Chemistry that has been taught by Professor Shagota Dasgupta of IIT Kharagpur. And I am Srijan Sen from IIT Kharagpur, and I have been taking your live sessions for the previous weeks, and we have come to the last one today. So okay, we can just start, and then we can see how far we can go with the discussion of the question and answers, and if participants are joining. Okay, so I'm sharing my slide. Okay, let us just wait for 2 more minutes, and then I think we can start if anyone else is eager to join. We understand that this is an extra class, but this class has been organized for you people only, so for your benefits and all. So we can go with the quiz session, but also if there are no participants, because these question and answers have been discussed, only a few questions that are very important for your exams needs to be discussed more, and just a small tips and tricks as to how you can go for giving an exam. So practice the numericals well, and there will be few direct questions as well as a few tricky questions, so try to answer them smart, because the questions that have asked you directly about those things and all, try answering them uh, in that manner. Okay, because don't try to proceed in a very tricky manner with uh, those, and solve the numericals well, because there will be 2 to 3 numericals most probably, not more, but try to answer them correctly. That will help you fetch good marks. So, in brief, if I just talk about the course details and all, so in the first week, we began with the introduction of the protein structures, and we talked about the peptide bonds. Okay, we talked about the amino acids, the peptide bonds, and how this peptide bond is forming. We talked about alpha helices, beta sheets. Then we moved to the protein architecture. Okay, so in the first week we talked about the titration curves and all. Then we moved on to the protein architecture where we talked about the Ramachandran plot. We talked about alpha helices, beta sheets, loops. Then we talked about protein folding and denaturation. Then how proteins can be isolated and how can you characterize them. Followed by how can you undergo these protein ligand interactions. When we talked about the protein ligand interactions, it also includes in the next following weeks we talked about your membrane proteins, then protein nucleic acid, protein interactions as well. Then we also discussed about the enzymes and enzyme mechanisms, their kinetics, inhibition. Then I guess we talked about protein, membrane proteins, transport proteins, and metalloproteins, motor proteins. You have the dynosines, the kinesins, and all. Then how can you determine the protein structure? So the structural analysis, protein structure analysis, and we ended with discussing a few special topics in protein chemistry. So that was the entire curriculum of this course. So it was a 12 a week course, and we are just about to end this course with this last session. And in the upcoming days you have an exam that is coming up, so study the lectures that were taught to you by the course investigator in details, and also if you need help you can contact me, because I have been taking your live sessions. You can contact me via mail, or yes, via the mail. So, or you can put up your questions in the discussion forum where their questions will be answered. So I think if anyone has to join, they can join later, but let's discuss about the questions. So these questions would basically are important. These questions have been previously discussed as well. So this is just a quiz type of a session, but anyway, so we can just carry on without others. So these questions are important for your exams. So study them well such that you can score good grades. Okay. So the first question starts with a diagram which says find which among the following combination of torsional angles is correct. So basically you have to find the phi, chi, and the omega. So as I discussed previously, that this torsional angle is basically the plane. Okay, so four atoms are basically involved over here. So if you see that it is relating to the backbone. So find the phi, chi, and omega. So if you see that the peptide bond, okay, so the peptide bond is your this particular one. So this one is being referred to. The next one is your C alpha C. So C alpha and C is your this one. Okay. And is your this one. Okay, so C alpha, and okay, so that is also a peptide bond, and this one. So yes, so basically you can find the correct answer. It is C, that your peptide bond is this one, that is your omega. This one is going to be your omega. This one is your phi, and this one is psi. This is also your omega. So it's a very easy question, and but try to remember this question very minutely, because this can be one of your questions even in the exam. Hello, Avisha. Hello, can you hear me? Okay. Let us move on to the next question. So, a peptide containing 85 amino acid residues with a molar extinction coefficient having a molecular weight of 10.2. If the peptide contains 4 tyrosine and the absorbance um, value of 0.393, then calculate the number of tryptophan and the concentration of the protein solution. The molar extinction coefficient is 1280 and 5690 respectively. So you have to find the number of tryptophans present and the concentration of the protein solution. So you see that the eta value is already given over here. Okay, so this is your eta value. That is given over here, and it says that you already have 4 tyrosines, so eta equals to 4 into this value, and plus x into 5690. So 4 into 1280 and x into 5690, which is equals to your 39260. Okay, 39,260. And from there you can get the value of x to find out the contribution that tryptophan has in this extinction coefficient, and you will get the value is 6. Okay, so when you get the value to be 6, from there you can calculate the concentration from A equals to epsilon Cl, and when you calculate it, A equals to epsilon Cl, where absorbance is given over here. This is your absorbance value, and you have epsilon to be this one, and L you can consider 1 centimeter and all, and you get the value to be 10 to the power minus 5 molar. You get 10 to the power minus 5 molar, that is equals to 0.01 millimolar. So this would be the correct answer. So these type of questions you can expect in your exams as well. Okay. Now, when we talk about the Henderson-Hasselbalch equation, so what message is this Henderson-Hasselbalch equation spreading? So Henderson-Hasselbalch equation does not explain the behavior of di and tribasic amino acid weak acids. Uh, okay, is useful with solution of acetic acid and hydrochloric acid, relates the pH of a solution to the pKa and the concentration of acids and conjugate base, and allows the graphic determination of the molecular weight of a weak acid from its pH alone. So if you see that the Henderson-Hasselbalch equation is for 3 only, where it relates the pH of a solution to the pKa and the concentration of the acid and conjugate base. So that is only your correct option. Okay. Now this is a titration curve. It is saying uh, at pH 8, which of the structure would you be observing? So which structure would you observe at pH 8? See, if you look into the titration curve at pH 8, the overall charge of the amino acid will be +1, as your pK1 is 2.1. So once the pH crosses 2.1 and until it reaches 9, the ionic form will be +1, and it would exist in that manner. So +1, here you have 0, +2, -1, and this is +1. So D is the correct option. Okay. Now, which property of myoglobin and hemoglobin, which property makes myoglobin and hemoglobin different? So is it the iron binding site, the large segments of alpha helix with the spiral shape? That is not correct. Oxygen saturation profile at a different pH, this is right. Cooperative binding of oxygen, this is right. Iron binding site, this is not correct. This is also not correct. So 1 and 2. So yes, so the section where we discussed about the myoglobin and hemoglobin, that is very important, and you can expect also one question from over there. Okay, now a hypothetical helix has 5.6 residues per turn with 5.2 angstrom pitch. What is the rotation angle per residue of that helix? So how you are going to do this to answer that? So you know that a circular turn is 360 amino acid, 360 degrees. So the answer would be 360 divided by 5.6. So it is 360 divided by 5.6. So you get the answer to be 64.3. Molecular weight of a polypeptide can be determined by. So how you are going to determine the molecular weight of a polypeptide? And that also precisely. So agarose gel electrophoresis is mostly performed for your DNA and RNA. We do that. SDSP can give you the molecular weight of a polypeptide, but not precisely. If the precisely term is mentioned, then you have to be very much precise with your answer as well. But MALDI mass spectrometry can give you the molecular weight precisely. So moldy MS would be your correct answer. Okay, the next question is primary structure of a polypeptide. So the primary structure of a polypeptide is formed by, it is a very easy question. So the primary structure is formed by the condensation reaction, where you lose one molecule of water to get one peptide bond. Which bonds do not contribute in the primary structure? That is also easy. In the primary structure, you have the NC bond, you have the SS bond, but hydrogen bonding and salt bridge interaction do not participate in the primary structure. So 3 and 4. The next one. Protein folding is a spontaneous process because why is protein folding a spontaneous process? It is a spontaneous process because your delta S of water, what happens in protein folding, there is a hydrophobic collapse of the polypeptide chain that leads to increase in the entropy of water. So your delta S water becomes positive and change in enthalpy is large and negative because hydrogen bonds, electrostatic interactions and your van der Waals interactions contribute to enthalpy. So the protein folding is mostly determined by or controlled by your enthalpy. So D is the correct option. The next one, dissociation constant has a unit of. So what is the unit of dissociation constant? Or you can tell KD. How do you get KD value? So you get it in say 10 to the power minus 6 molar, 10 to the 1 nanomolar. You tell that the value is 0.35 micromolar. So you get it in terms of concentration in molar, nanomolar, micromolar, millimolar, and all. And lower the KD value, higher is the tighter is your binding affinity. Collagen is formed in connective tissues such as tendons, cartilages. So collagen provides the strength due to. So what is collagen? Collagen is a so collagen is basically a coiled protein, and in the coiled coil there are three separate polypeptides known as alpha chains that are super twisted about each other. So alpha helix is responsible for the strength of your collagen. Now coming to the next one. A protein without an affinity tag is being purified from sonicated crude extract. Which is the best purification technique? So it does not have a affinity tag. So if it does not have an affinity tag, it cannot be purified by nickel NTA. So A and C is incorrect. The other option is ACC, ion exchange and salting out. And salting out, ion exchange and ACC. So D is the correct option. First you salt out, then you do an ion exchange, and lastly you run it in ACC and perform it. The three-dimensional structure of a protein can be determined by how can you determine the 3D structure of a protein? Okay, so for that you can perform NMR and extracrystallography. Okay, you can get the three-dimensional structure of a protein. The next one is particle size and sample homogeneity can be determined by. So it's a direct question, sample homogeneity. So you can perform DLS to check the particle size and the sample homogeneity using the DLS technique. Which among the following statement is correct? Soluble proteins have high density and low sedimentation coefficient. Nuclei have lower density and higher sedimentation coefficient. RNAs have lower density and higher sedimentation coefficient, and mitochondria. So the only correct option over here is 2. Nuclei have a lower density and higher sedimentation coefficient. The next one, which of the following statements is or are correct? In dialysis, molecules diffuse through semi-permeable membrane if smaller than pore size. That is correct. Dialysis is used to remove low molecular weight compounds. That is also correct. And in ultracentrifugation, molecules migrate through a semi-permeable membrane under pressure. That is also correct. Ultracentrifugation can be used for crude fractionation and buffering. No, that is not correct. So A is the correct option. The next one, IPTG. This is a question regarding IPTG, and this is a very important question for you people. So IPTG. So what is IPTG? What is not true about IPTG? There is a molecular mimic of allolactose. That is correct. Oh, find the not true. It's not true. So part of the metabolic pathway. That is not incorrect. So 2. Okay, so we have only 2 and 1. So 1 is correct. So this one will only be the incorrect option. Okay, now the next question is, you know that the protein you want to purify from a natural source forms a multimer. Okay, so it forms a multimer. Okay, so what can you do? So small amount of target protein in the total protein sample. Which of the following is an appropriate purification strategy? So the molecular weight of the protein in solution suggests that the ACC would be appropriate because it's a multimer. However, the filtration properties of ACC matrices compared to the capture properties of your ion exchange and affinity chromatography means that only small sample volumes can be loaded on the ACC columns, say typically 1% of the column volume. So, as your target protein is already dilute, an unreasonably large column would be necessary for purification of a useful amount of protein. Now, ACC also dilutes samples further during chromatography, so capture steps such as ion exchange affinity chromatography can be used to be purified from a natural source. So, therefore, you can basically. But now the concept of affinity column used over the ion exchange column comes. So ion exchange chromatography is a better capture technology than affinity chromatography. So you can use that before using ACC. So IEX followed by ACC. These were the questions that were discussed to you also. So you people can do it. These are easy questions. These are quite easy questions. So what do you use EMSA for? You use EMSA for protein nucleic acid interaction. Ligand A and B binds with the protein with a dissociation constant of 1 nanomolar and 100 nanomolar. Which of the following is true? A binds P more tightly. Yes, because lesser the one. So A binds P more tightly. The protein binds the ligand with a dissociation constant of 10 to the power 6 molar inverse. What is most likely to be the KD? The presence of a competing inhibitor. So the KD has to be in molar and negative, so it reduces. In presence of a competing inhibitor, it will just increase a bit. Okay, so it becomes 10 to the power minus 5 molar. Now in fluorescence anisotropy experiment, a small ligand is stacked with fluorescent label. So the polarized label is excited by the polarized light. An increase in the rotational diffusion of the small molecule is detected. No, the decrease is detected. So the decrease is detected and the fluorescent label is being polarized. So BNC is correct. Okay, surface plasmon resonance detects the change in heat after the change in refractive index, the change in migration of the protein. It's the change in the refractive index. Not true for an isozyme. Okay. Now what is not true for an isozyme? So basically your isozymes are enzymes that differ in the amino acid sequence but catalyze the same chemical reaction. Okay, so they can be, they do not have the same km values. This is also true and regulated differently. Isozymes have different kinetic parameters and they are regulated differently. Yes, that is correct. So but only they have different km values. That is not true. Okay, for a reaction where SEP stands for substrate, enzyme and product respectively, the quantities that can be measured experimentally are, so what can you measure experimentally? You can measure the total enzyme, substrate and the product, all three of them. So which of the following determines the substrate specificity of an enzyme? So the complementary geometry of the substrate and the enzyme, complementary electrostatics of the substrate and the enzyme, and the degree of hydrophonicity. Yes, so these two are correct. Let's move on to the next question. For competitive inhibition, the maximum velocity can be obtained at. These are the questions that are direct questions you can do on your own, so I'm not discussing, but just look at them. These are direct questions, you can find the answer. What I need to talk about is the Michaelis Menten. Okay, just a minute. Okay. Okay, now from the Michaelis-Menten kinetics, what can you actually derive? So there are multiple substrates. Substrate binding is irreversible. Uh, K2 equals to Kcat. The enzyme displays cooperativity. So if you check, so in Michaelis-Menten kinetics, K2 is not equals to Kcat, and there are multiple substrates. The substrate binding is irreversible, and the enzyme displays cooperativity. So 1, 2, and 4 is correct. Okay. The next one, in competitive inhibition, an inhibitor binds. So an inhibitor binds reversibly at the active site. So it binds at the active site. Now the reaction shows something, formation of an enzyme substrate complex followed by the release of the product. So the rate of ES breakdown will be. So if you check, the rate of ES breakdown will be the first one. K minus 1 into ES, K2 plus K2 ES. This one is about okay. Now match the following. The role of polymerase, helicase, guidase, and the ribosome. So what does the polymerase do? So polymerase catalyzes the phosphodiester bonds between the nucleotides. So one is D. Your 2 is helicase that separates two annealed nucleotide acid strands. Guidase catalyzes DNA supercoiling and relaxation, and ribosome reads and translates mRNA to a polypeptide. These questions are very scoring questions if you know just one answer. See, I just looked at the polymerase that catalyzes the phosphodiester bonds between the nucleotides. I got the entire option to be correct. Okay, so in this manner you can solve a lot of questions. How many else do I have? I have quite a few questions. Okay, we can take a break of just 5-10 minutes and then we can start again. Okay. Okay, so let us start with the next set of questions. So this question is about your azurine. So let's see what it says. Just 2 more minutes. Just Yeah, sorry for the delay. Okay, let's see where were we. Okay, now we were speaking about azurine. Okay, so when we speak about azurine, so we have to know that azurine is a tetrameric protein having four copper centers, and these copper centers are coordinated by two N-histidines, three cysteine residues at the equatorial position, and one carbonyl oxygen of your glycine and methionine. So therefore it has two carbon, so false. So it has four carbon ions, so that is false. So 3 and histidine and 2 cysteine residues. Basically it has three cysteine. Yeah. So therefore 1 and 3 is the incorrect option. Next, catalase, peroxidase, rubridoxins, and ferredoxins. So the work of catalase is catalyze the disproportionation of H2O2. Peroxidase oxidizes organic substances by H2O2. Rubridoxins does not have inorganic sulfur, and ferredoxins have multiple centers. Okay. Hemocyanin. Okay, false statement about hemocyanin. So each copper ion in hemocyanin is coordinated by 3 N-histidine residues, not 2. So that's a very direct question. Okay. Now, which statement is false regarding myoglobin and hemoglobin? It is false about hemoglobin and myoglobin. Oxygen saturation is higher at low partial pressure of oxygen for myoglobin. That is right. Saturation is higher at low pressure. Okay, so I discussed this previously, so you can find out the answer from my previous one. Okay. Which of the following is or are true? Dyneins are retrograde transport, that is that is true. Kinins are retrograde transport, no, that is wrong. Kinesins help to move the chromosome along the microtubule, that is right. So 1 and 3 is correct. Which of the following statements is or are indicative of passive transport? For passive transport. So if you read, you can understand that only small organic molecules will diffuse based on molecular weight and solubility in lipids, and the other correct option is molecules are transported from high concentration to low concentration. So D is correct. Actually, we need to have participants to be more interactive, otherwise it becomes uh, you do not get the excitement of a quiz session. Okay, never mind. If the substance is uncharged, what information would you need to predict whether the transport will be thermodynamically favorable? So, information about the concentration gradient. Yes, that is correct. Now, indicate whether the following compounds are likely to cross the membrane by channel or by transporter. This is a very interesting question. So, if you see that calcium and water can pass through the channel, but you need a transporter for glycine and glucose. The next one is a problem that you have to solve. So where you have to use delta G equals to your RTLN, sodium means ion concentration outside divided by ion concentration inside, plus you have the ZF delta E. Okay, and when you calculate, from there you have to calculate delta E. So RTLN ion outside by inside equals to minus ZF delta E. From there, if you calculate, you get a value of minus 75.7. So all aerobic organisms, so all aerobic organisms require oxidative phosphorylation to produce basic energy by generating ATP by an ATP synthesizing enzyme called ATP synthase through a process called electron transport. Now solid band, you observe a solid band for pi to pi star and transition for categorizing of heme prosthetic groups. Okay, so DNA binding tetrameric protein. So see, a lac repressor is basically the DNA binding tetrameric protein having four identical subunits which operates in by a helix turn helix motif. In the DNA binding domain, it inhibits the expression of genes coding for proteins involved in the metabolism of lactose in bacteria. And this bound repressor can reduce the transcription of the lac proteins by occluding the RNA polymerase binding site or by prompting the DNA looping. So based on that, the last one is correct. It is a lac repressor protein, functions by prompting DNA looping obstruction polymerase binding site. Okay, now find out the lectins. So find out the lectins. Okay. So if you see that for cell addition you have the C type, for the development and maintenance of nervous system you have P type, and for roles in generation of functional lysosomes you have the I type. So these questions are direct questions, nothing to discuss more. So what is not true about the RNA footprinting assay? So the region that is protected is known as the footprint of the protein. That is not protected is not the footprint. The region that is protected is the footprint. And proteins bind to RNA sequence that can protect the region of RNA, that is right. It makes it possible to locate. The other options are correct. So now another match the following. SRC homology 3 SDM limb. So your SRC homology 3 binds to proline rich sequences. So 1Y. Okay, 1Y. So C or D. Then your SDM binds to non-SAM domain containing proteins, also RNA. Yes, SDMW. So it is D. And your limb is binds to helix loop helix motifs, and your PDZ is recognizing the STXB motif. The correct statement about protein corona finding nanoparticles. So the soft corona is formed by nanoparticle bound protein-protein interactions. That is correct. And the hard corona is formed by nanoparticle protein interactions. So 1 and 3 is correct. So identify the proteins from the features. Belongs to a large family of molecular chaperones. So chaperones means heat shock proteins, key role in protein maturation, refolding and degradation. Okay, involved into stressful conditions, exposure to high temperature and signal transduction protein. So it is your HSP90. Peptidometrics are used for. So what do you use peptidometrics for? So peptidometrics are used for, yes, binding of tight sequence, modification of the peptide to enhance stability, delivery and upgrade, development of drug-like compounds from existing peptides. Yes, all three are correct. Now, staple peptides bind to estrogen receptor using a hydrocarbon link to stabilize them in the form of an sheet, random coil, turn, and an alpha helix. Yes, that's an alpha helix. Correct. Which of the following is or are correct about the SRC homology domain? Okay, that's a good one. So, an SRC homology domain is one of the two small binding domains found in the SRC oncoprotein, and it is a non-receptor tyrosine kinase protein which is encoded in humans by the SRC gene. It includes an SH2 domain, an SH3 domain, tyrosine kinase domain. So it's not a tryptophan kinase domain, it's a tyrosine. And activated SRC regulates the activity of matrix metalloproteinase. So 3, this one is incorrect. Okay, so we have last 3 questions. So which of the following processes do not occur in HIV? So the process that does not occur in HIV, and the right answer is replication and transcription, because in HIV reverse synthesis of DNA from RNA is performed by a reverse transcriptase. So translation is the synthesis of proteins from mRNA. Since mRNA carries instructions for making new viral proteins, each section of the mRNA relates to making a different part of the HIV. So C is. Which among the following is not true about IDPs? So IDPs carry high densities of polar charged amino acid residues like lysine, glutamine, serine, arginine, and glutamic acid. So it contains high density, not low density. So the last one, tryptic digest. So if you see that I discussed this quite a few days back only. So that the cleavage is not possible when there is a proline following it. So this is a tryptic digest by trypsin. So it will cut after the K and N. But you see that there are 2 tripeptides and 1 dipeptide. And both the tripeptides does not contain arginine. So the arginine. But there are 3 cut sites, 2 for lysine and 1 for arginine. So there should be 4, but you are getting 3. That means there should be 1 lysine or an arginine that is following a proline, that it should not cut. Okay, so for that, let us see where is a proline following your lysine or arginine. So proline following lysine is over here and here. So these 2 are incorrect, and see you are getting, if you cut it you get over here 1 2, and here you get 1 2, but your tripeptide does not contain arginine, so this one is correct. So that's all then. Best of luck for your exams, and answer the questions very calmly, and think before answering. There will be direct questions for you as well, so don't try to make it more complex. So try answering them in a direct way, and don't get confused, because there might be a few questions where you might get confused with the answers. Look whether it is an MSQ or an MCQ, and then answer it in a calm head. So best of luck to all of you for your exams, and I hope that you get very good grades in that. Okay then, thank you all, and if you have any queries you can just send me a mail in my email ID. Okay, I'm texting it over here, so you can send me a mail, I will answer you. Okay then, have a good day, and thank you.